Nicotine is the addictive component of tobacco products and e-cigarettes. Based on what you know about the acetylcholine receptors and their widespread distribution in the peripheral and the central nervous systems, you can probably predict that nicotine has a lot of effects. Nicotine accomplishes these effects by binding to the nicotinic acetylcholine receptor. It's an agonist at these receptors. Now remember that there are multiple subunits of these receptors, and the specific combination of subunits actually changes the affinity of nicotine for the receptor. This means that nicotine doesn't bind to all acetylcholine receptors equally, and there are certain types of acetylcholine receptors that primarily mediate the effects of nicotine. Remember, there are two different types of the neuronal nicotinic acetylcholine receptors. The alpha-4 beta-2 receptors contain alpha-4 and beta-2 subunits, and these have a high affinity for nicotine. Nicotine binds very easily and readily to these receptors. On the other hand, the neuronal alpha-7 receptor, which has 5 alpha-7 subunits, has low affinity for nicotine. Nicotine does bind to both of these receptors, and they both can influence the effects of nicotine on the brain. These alpha-4 beta-2 high affinity receptors are located in the hippocampus and in the ventral tegmental area and in the cortex. And nicotine has a wide range of effects in those areas. Let's take a look now at how imaging studies have allowed us to visualize this nicotine binding to these nicotinic acetylcholine receptors in the human brain. What we're looking at here is a combination of MRI and PET scans of the human brain, and these are in smokers who are abstinent for a couple days from nicotine. So here we can see just the structural images of these brains. And this is done using MRI, right? So MRI is very good at giving us spatial localization and giving us these nice pictures of the pathways and areas of the brain. And what they've done then is combined this with PET scans. And PET scans are very good at allowing us to see binding to various receptors or different proteins in the brain. So what they do in this case is they give a radioactive ligand that binds to the nicotinic acetylcholine receptor. So you can see here, these are when the smokers are abstinent, so they haven't been smoking for a couple days. And we're just looking at 3 different planes of interest in the brain, so different regions. This top one here shows us really nicely the striatum, which you can see structurally is right here. The striatum is really big in the middle of the brain. And we can see that any red is going to be a high amount of radioactivity. So this is a radioactive ligand that will bind to the nicotinic acetylcholine receptor. So this shows us that in the striatum we have a high level of expression of the nicotinic acetylcholine receptor because a lot of this ligand is binding and giving off radioactivity in that area. Now, what happens if these individuals smoke? In this experiment, they gave them a denicotinized cigarette, so it doesn't have very much nicotine at all, a little bit, and a low nicotine cigarette. What we can see in this lowest nicotine level condition is that in the striatum, we see a change in the amount of binding of the radioactive ligand. So after the participants have smoked this denicotinized cigarette, the amount of receptor that's available to bind to this radioactive ligand has decreased. We're seeing less binding to the radioactive ligand. And that's because nicotine is now in the system and has bound to the nicotinic acetylcholine receptor. So the receptor is now occupied by nicotine and there's less of it available to bind to the radioactive ligand. So what we see is this decrease in the amount of the radioactivity, which shows us that nicotine is binding to receptors in these areas. And in fact, in the low nicotine condition, which is more nicotine than in the denicotinized condition, we see even more binding. So you can see all throughout the brain, and especially in the striatum, these big changes in the amount of receptor that's available to bind to the radioactive ligand. And so this tells us that nicotine is binding to receptors all over the brain, but particularly in these areas where these high affinity alpha-4 beta-2 nicotinic acetylcholine receptors are expressed. In naive users, nicotine tends to produce feelings of tension and arousal, as well as feelings of nausea and headache and dizziness. This is due to the effects in the peripheral nervous system as well as the central nervous system. Remember that acetylcholine is involved in arousal, and we see this when nicotine is taken. It can induce feelings of heightened arousal and attention. Now, in individuals who are actually dependent on nicotine, so for example, people who are heavy smokers, nicotine tends to produce a state of calm and relaxation. And a lot of this has to do with the fact that those individuals are experiencing relief of a withdrawal syndrome. So withdrawal causes feelings of anxiety and etc., and that can be relieved by administration of nicotine to smokers. So we have to be careful about when we think about the effects of nicotine. Is it really a relief of withdrawal, or are these the acute effects of the drug? We also know that nicotine can increase attention. This has been demonstrated in both animal models and studies with humans. Again, in smokers, this is true because individuals in withdrawal have trouble with concentration, and so giving them nicotine and relieving the withdrawal can improve attention. But it's also been shown in naive individuals. And of interest, it's particularly been shown that individuals who score lower on attention at baseline seem to see particular improvements. This is demonstrated in this graph, where this is a stimulus detection task, which is one way to measure attention in humans. Basically, are they paying attention and can they detect a stimulus on a screen? And you can see they've grouped their participants based on whether they had low attention or high attention at baseline. And individuals who score low on measures of attention actually do better after they've been given nicotine versus a placebo, whereas there's no difference in individuals who already score high on attention. So this demonstrates that nicotine can improve attention in humans given the right study conditions. Nicotine is also reinforcing, and we know this in part because nicotine is one of the most addictive drugs. Tobacco products and e-cigarettes are highly addictive, and people often find it almost impossible to quit using these drugs. We also can see in animal studies that animals will self-administer nicotine under the right conditions. This is an interesting point about nicotine, actually. Because it can be aversive when first administered, remember those feelings of headache and tension and nausea that we talked about? Animals actually won't readily self-administer nicotine, and they can actually become very irritable upon administration of nicotine. But if the experiment is done right and you can overcome some of those negative effects, it has been shown that animals will self-administer nicotine, demonstrating some of its reinforcing effects. Now, nicotine is thought to be reinforcing for a number of reasons, but one of the most important is the activation of VTA dopamine neuron firing. So what we're looking at here is the firing of a dopamine neuron located in the ventral tegmental area. And each of these lines here represents a spike. So at baseline, VTA dopamine neurons fire sort of intermittently. And what we see is that when nicotine is given, we get these phasic bursts of firing where a number of action potentials are fired in rapid succession. And this again occurs because remember those acetylcholine neurons that project from the pons to the ventral tegmental area. And there are nicotinic acetylcholine receptors, the high affinity kind, on these VTA dopamine neurons. So when nicotine is present in the system, it's binding to these receptors and causing activation, depolarization of the neurons, which triggers this phasic bursting of the VTA dopamine neuron. This is important because it means more dopamine is released in the striatum, and we know that dopamine is incredibly important in the manifestation of addictive behavior and in reinforcement. In addition to the effects that nicotine has directly on the cell bodies of VTA dopamine neurons, we also know that nicotine can influence the axon terminals of those same dopamine neurons. So those VTA dopamine neurons, we'll learn this in more depth later, but they project from the ventral tegmental area to the striatum, and that's where their terminals are, where they release dopamine onto cells called medium spiny neurons. So we've already discussed these projections here from the pedunculopontine tegmental nucleus. These are cholinergic neurons which send projections to the VTA and release acetylcholine onto the dopamine neurons. The nicotinic acetylcholine receptor then binds to that acetylcholine, and so nicotine can influence dopamine neuron firing through that mechanism. But in addition to this, there are interneurons that are cholinergic that live in the striatum, and they release acetylcholine onto the axon terminals in the nucleus accumbens and the dorsal striatum. And they're very important, we're learning, in controlling the release of dopamine in the striatum. And so nicotine has the potential to influence firing both at the cell body of these VTA dopamine neurons as well as at the axon terminal. So here we've zoomed in on the terminals of those dopamine neurons, so the VTA dopamine neurons that project to the striatum. These are the terminals. And they synapse here onto these medium spiny neurons. These are actually GABAergic neurons. The majority of the cells in the striatum are GABAergic, and they project, so they're GABAergic projection neurons that have inhibitory influences over other brain regions. And these cells release dopamine onto the medium spiny neurons. But today we're focused on nicotine and acetylcholine. So let's focus here on the nicotinic acetylcholine receptor, and you can see that it's expressed on the terminal of the dopamine neuron. Now remember, these are ionotropic, excitatory receptors. So these cholinergic interneurons, that means they have their cell body and their terminals all within the striatum, they synapse onto the dopamine terminal. They release acetylcholine, it binds to the nicotinic acetylcholine receptor, and this produces positive current in the terminal. And through this mechanism, these cholinergic interneurons can regulate dopamine release. So nicotine can obviously bind to the nicotinic acetylcholine receptor and influence dopamine release in this fashion.